So today I'm just talking about a study that is still ongoing. Data collection is still proceeding, and we're also undertaking analysis on this study. And the study is on the dorsal tongue microbiome of human subjects in Singapore, which is a subcategory of the oral microbiome. And some acknowledgements here. My colleague and collaborator, Charlene Goh, who's an assistant professor at NUS Dentistry, is the lead PI on this study, and we are, Mindia Hariano is the lead analyst, and we're collaborating with Charlene and her colleagues from dentistry and from NUS Medicine and NUHS on this study, which was funded by just internal startup funds from NUS, from CELSI, and from the NRF and MOE. Sorry, the NMRC and the MOE under the AIF program was funded the background study. And disclosure, I'm a co-founder and equity holder at a microbiome anchored biotechnology company in Singapore called Blue Maiden Biosciences. And so, so we had just had a very nice talk from the previous speaker about microbiomes, or the fungal component of human microbiomes. And if we think about microbiome, or microbial communities, I mean, complex microbial communities is a more general term that is used to describe microbiomes in the context of microbial ecology. They're really a set of problems that drive work in the field, and fundamentally most of the techniques that we're talking about were developed because we don't know much about the member species of such communities, be they bacteria or fungi, viruses or microeukaryotes, and we can't culture and can't isolate and culture most of them very, very easily. And so this necessitated the development of so-called culture-free techniques typified by metagenome sequencing. Even if we did know what these species are, and we know increasingly large swathes of prokaryotic phylogeny, we don't really know much about what these species are doing, and nor do we know much about how they're contributing to health and disease. And so there's various ways that this can be addressed. New culturing techniques is a really important area in any microbiome or complex microbial community analysis, as is developing new ways of analysing the genomes of these species and new ways of studying their function in situ. And I think many of you will be familiar with this sort of workflow. There's a microbiome sample that's obtained from some site, in this case in the gastrointestinal tract. The DNA is extracted from this sample and then sequenced using high throughput sequencing technologies. And then there are various things we want to know about the microbiome. We want to know something about the genome content of the member species. We want to understand, we want to understand, the nature of individual genes. We want to know what kind of species are present in the microbial community and what their functions are. And there's several ways that we can go about this. The two major approaches are either to treat the reads themselves as the unit of analysis, and sort of try and infer where those reads are coming from and what genes they're coming from. Or we can assemble those reads together into contigs and scaffolds, and then try and formulate working genomes of the member species. And so in this case, we're studying the dorsal tongue microbiome, which is the microbiome that inhabits the sort of upper, or the dorsal, the upper surface of your tongue. And this is actually the microbiomes of the oral cavity are actually quite relatively distinct from one another. So you'll have a different sort of microbiome on the inside surface of your cheek, and you'll have a different sort of microbiome again on the surfaces of your gums, or the interface between your gums and your teeth. And also the saliva microbiome is kind of related to each of these as well. And they're obviously not completely distinct communities, but they show some systematic variations in data that's been conducted to date. They also, species in these microbiomes also have a rather distinct spatial morphology, which is obviously not addressed by any techniques I'll talk about today. And also a reminder that, as we heard in the last talk, it's not only bacteria, fungi and viruses are also an important part of these communities. And I think we're used to, there's a certain, we're used to hearing a lot, I think, about how microbiomes are important for everything now, and that's, that's kind of the type of hype you expect from a new field, or a relatively new field. And there's I think that there's a lot more skepticism and critical thinking should be applied to these sort of problems. But it's pretty clear that there's been a long-standing link, for example, between the microbiomes of the oral cavity and cardiovascular disease. There's a lot of emerging data on the importance of species that are transmitted down the gastrointestinal tract and their importance in disease such as IBD. Respiratory, the link between microbiomes of the oral cavity and the respiratory tract are important in certain types of populations, and there's also other strong data emerging about the links for other diseases such as neurodegenerative diseases and diseases like rheumatoid arthritis. And so this study population arose from the study that was called Assessing the Progression of Type 2 Diabetes, or APT2D, and this was a study that was started and led by Suan To, who was then at NUS and NUHS. And it was about 2,500 subjects. There was a sub-cohort of this sub-cohort study that was conducted, or is being conducted by Charlene Go, in which we are collaborators for the microbiome analysis. That took dorsal tongue samples from about 500 individuals in this cohort. There's an extensive set of phenotype data that's available from the background cohort, and there was Illumina sequencing performed, and currently we're in the process of collecting a subset of samples using nanopore sequencing, which we only have 3 at the moment, but I put this in here because we have indeed collected them, and more are underway. And then there's various types of read level and assembly level analysis that have been conducted on this data that I'll briefly summarise some of that today. If we look at the most basic way of looking at this, the richness or just the overall complexity of these communities are actually quite low. You'll see these numbers a lot in microbiome analysis. They're called ecological diversity indices, and they can be sometimes hard to interpret. I find it a lot easier to think about something like this, which is if you just look at the number of taxa that you need to capture 50% or 75% of the total relative abundance, and depending on how you depending on what profiling technique you use, but if you use Metaplan 4, for example, you can see these are relatively simple communities. You have about 15 or 20 species that are giving you, about half the total relative abundance, and after that you have a much lower abundant tail in the distribution. Many of these species here are known to be associated with the microbiomes of the oral cavity. Some have disease associations as well. And then if we look at the overall community structure, and this is a sort of ANOVA type analysis where you're looking at sources of variation and how that affects the structure of the intersample distance matrix. This is the effect size here on the horizontal axis, and this is a p-value adjusted for the number of tests, and we can see that salivary pH is the most dominant effect here, but it's only really accounting for less than 3% of the total variation in the community structure. Age, BMI, ethnicity, and the degree of health of the gum are also important factors. But everything else is sort of less remarkable in the effects. And this is kind of consistent with what we see with other studies. there's a very, very high degree of inter-individual variation that is associated with the structure of these communities, and these kind of systematic effects are generally accounting for relatively modest amounts of variation. And so one thing we were interested in many other people in this field are interested in doing is constructing systematic catalogues of genomes from these communities and comparing those genomes across, within microbial groups. And so to do this, you're using the same data, of course. You've got the reads here, you assemble them into contigs, and then there's various kind of statistical or ML type procedures that are used to cluster those contigs together into working models of the genomes of the species in the community. And there's an increasing diversity of these approaches that are being generated. Metabat2 is one of the oldest ones and still one of the sort of best performers, but there's a whole suite of new ML or deep learning based approaches. This one, VAMB, is a variational autoencoder, which we've been also been working with. And the aim here is that you basically analyse you're using information about contig coverage and also about the contig sequence properties, like essentially K-mer distributions, and you're combining that information together and then clustering out kind of putative working models of the genomes. And there's a sort of understood, there's an accepted framework for defining quality of these putative genomes. And as you can see, irrespective of what technique you use, and this is typical for results in this field, most of the results you get will be pretty unremarkable. Most of the genomes will be of medium or low quality results. But between these two techniques, you see a big difference in this fourth category, which are basically genomes that are complete, but they're very, very badly contaminated, and that they're probably containing multiple genomes of closely related types, or maybe multiple strains together. And VAMB is interesting. The variational autoencoder technique is interesting because it's controlling this contamination very, very well, albeit the trade-off is that you don't get as many genomes, but the ones you get seem to have a better quality. If you look at this a bit more, this is these four categories of quality again, and we're just plotting the number of contigs in the genome. And you can see, of course, that some of them are very, very badly fractionated. You have hundreds or thousands of contigs that are forming these genomes. You can see a kind of left shift here in VAMB compared to Metabat. And I think this is actually really, really important information about how VAMB is actually controlling contaminants from non-cognate genomes in the analysis when you're looking at a given genome, and I think that there's the phenomenology of that deserves to be explored a lot more. Finally, what in terms of the taxonomic novelty, are we picking up a lot of genomes in this analysis that are genuinely new, or do they recapitulate genomes that are already known? And depending on how you look at this, if you use the GTDB is essentially a new initiative in culture-free microbiology to basically make a bacterial phylogeny or perform bacterial systematics based on genome data, driven by Phil Hugenholtz at the University of Queensland. And until relatively recently, doing taxonomy on these genomes that you would recover from these analyses was quite difficult. You either had to have very close reference genomes, or you had to extract the 16S gene if indeed it was assembled properly, which most of the time it was not, and then use that to infer the technology of the genome. And that's changed radically in the last couple of years, and you can see here that in these samples, there's about 90% of the over 90% of the genomes we recover are classifiable at genus level, or about half that at species level, which is quite a good yield. Interestingly, actually, if you just compare against these catalogues of genomes people have assembled from from oral microbiomes, you don't get a very high recapitulation rate, and I think that speaks to the strength of the GTD project in terms of defining a useful bacterial phylogeny. So I can make a few conclusions on this preliminary analysis. So data analysis is certainly ongoing for this, but also data collection, and I think a lot of the long read data we get will make a big difference to our ability to recover these genomes. But as with other microbiomes, the composition and the community structure of these microbiomes seems to be subject to a very, very high degree of inter-individual variation. There is no sort of obviously dominant environmental or clinical variable that's driving the structure, although that may benefit from more detailed covariate analysis and other ways of looking at this. And we've seen this multiple times with the VAMB workflow, but it appears to control contamination really, really well. And I think this is a really, really important issue in the analysis of metagenome assembled genomes, or MAGs. In part because I think the assessment of quality in these genomes has become very canalized. It's become very dependent on the use of so-called single copy marker genes. It's a very, very useful framework, but by nature it's an imperfect framework, because you might have 100 contigs in a genome, and maybe only sort of 15 or 20 of them will actually contain one of these marker genes that's used to define completeness and contamination. So I think if you're working in this space or you're getting involved in it, I think to some degree we might have over-metricized what constitutes a good quality genome relative to the complexity of actually defining one. And at the end of the day, that's the important thing. It's not whether you've got 92 high quality genomes or 85, it's actually what those genomes are like and whether they're useful to a broader audience. Although the taxonomic specificity seems to be quite high, I think this is probably very dependent on quality, and this is related to my previous point, that if we're going to undertake a lot of comparative genome analysis, you actually want to have high quality genomes if you're going to make inferences about things like, for instance, regions that differ between one major strain and another, major subspecies and another, for example. What I haven't mentioned today, we are in progress analysing other components of this microbiome. Sorry, I'm sure everyone's completely jaded with special issues, but this is a Royal Society of Chemistry journal called Molecular Omics. It's not for profit, which I think is an important thing to support, or at least balance against other types of scientific publication models. This is a current open call for papers on multi-omics and microbiomes. The editors are myself and Sean Gibbons, Anna Kachina, and Kong Ning from Wuhan. So please have a chat to me if you're interested in submitting a paper. And thank you very much. I think we're in time for lunch.
Thank you, Rohan. Time for questions. Let me ask a question.
Everyone wants lunch.
Yeah. So you showed, you look at the proportion of the microbes in your tongue, and you listed the top 20 just now. So among these top few, I don't know, top 20 or 100, how many of them remain stable over a period of time? Or there are some that will come on and off. I mean, they go high ranking and then after that low ranking. Is there some, stochastic response there in terms of the community?
Yeah, I mean, there's no longitudinal. There is a bit of a subject repeat data, like retest data in this cohort, which we haven't yet examined in detail. I think it's very variable depending on what. How your environment's changing. There are really nice studies, for example, that show that in the gut microbiome, at least, if you take a radical perturbation, like if you go and live in another country for 6 months, yeah, your microbiome will change. That's absolutely clear. When you come back to your home environment, your microbiome will return to roughly what it was before. So there certainly is a degree of plasticity there, but you know, it's probably a little more stable than people think. Yeah.
Thank you, Rohan. Oh, 2 questions, sure.
Stephen Brenner from University of California, Berkeley. So you mentioned briefly about association with disease, but I didn't see in detail what diseases you were looking at. And I was wondering, are you looking at systemic diseases? Are you looking at local diseases? Are you looking at things like geographic tongue, which is sort of the main thing I think of as being associated with the tongue?
Yeah, so this isn't a the cohort that this study comes from is a pre-diabetes cohort. So there are people across the normoglycemic to sort of diabetic, early stage diabetic range without frank diabetes. There's a few people that convert to full-blown diabetes, but that's there's no, at least in this in this case, there's no frank association with that clinical axis. That effect is quite modest, at least to the extent it's been analysed at the moment. There are other data about, I mean, the degree of periodontitis is a reasonably strong effect. So we would anticipate that would there'd be a lot of future disease trajectories that would come off that. And there's no, we're not doing any sub-analysis of the geography of the dorsal tongue. That's beyond the scope, although a very interesting area.
So actually, I was referring not to subgeography of the tongue, but actually a geographic tongue, which is one of the most common sort of traits that differentiates tongues.
Okay. Sorry, we're talking geography in terms of the tongue geography or human geography.
Geographic tongue it's not exactly a disease, but it's like a disease of the tongue.
Oh yes, geographic tongue. Yes, sorry. Yes, now I know what you. I'd have to check the phenotype data. It may be in there. It's quite an extensive collection, and a lot of that data is still feeding in from the host study.
Great, thank you.
I have one question. Have you detected any genes or genomes that belong to viruses or fungi? Because I suspect there might be some non-bacterial genes or genomes. So have you checked?
I mean, there will definitely be. There will definitely be non-bacterial content in that assembled sequence. There's some really nice new workflows for categorizing contigs as to what they are, whether they're really broad scale categorization, whether they're prokaryotic, eukaryotic, whether they're plasmids or viruses. So we are running everything through a couple of those at the moment.
Okay, can I ask one more question?
Sure.
Are you planning to compare your results with data from other regions, like from the public data?
Yeah, so the comparison to HOMD, the Human Oral Microbiome Database, that's like an aggregated data from many studies, but there are some really big oral microbiome studies that have been performed in China, which I think the mags are available for one of those studies. So yes, definitely.
Okay, thank you.